everyone. Just move this down. My presentation is on using vaccine immunostimulation, immunodynamic modelling methods to inform vaccine dose decision-making. So first, I'm going to outline exactly what I mean by immunostimulation, immunodynamic modelling, because this is a term that we have generated ourselves to try and reflect the fact that what we're trying to do is use PK/PD methods on vaccine data. So immunostimulation, IS, stands for the immune response stimulation following vaccination, and the ID part stands for the measured immune response dynamics as a result of this simulation. So as a kind of a starting point for doing this work, we looked at dose response data in humans following the H4 TB vaccine. And it shows that if you look at the data which I show on the right, that there is a non-saturating dose response curve. Particularly if you were to look at day 56, you would see that the 5 and 15 microgram doses are superior in terms of an interferon gamma response to the 50 and the 150 microgram of the vaccine antigen, with the adjuvant kept constant. And this is not a unique phenomenon. This has been seen in other diseases, for example, in malaria and in flu, so this isn't something that we aren't already aware of. However, for the next safety studies, a 50 microgram dose was taken forward. So which dose should we be choosing to study from this vaccine? Have we been choosing vaccine doses that have been too high, or should we have been going with the lower ones, for example, 5 micrograms? So our idea was to say, okay, so can we take the animal data, the animal dose response data, and mathematical modelling to predict what range we think we should be testing in humans? So very much that translation from animal to human. So the aim here, use ISID modelling calibrated to mouse multi-dose response data from the novel TB vaccine H56 to predict the human dose response dynamics. So we did this in 2 stages. The first one was to generate longitudinal H56 dose response data in mice, and then look at the dose response curve to see if we can finally say, we definitely cannot see a saturating curve here, this is definitely something else going on here. And we fixed that in different time intervals, and we assessed the dose response curve at these different time intervals. And then the second stage was to translate the longitudinal dose-dependent responses from mouse to human. So I'll start with stage 1. So we went into the lab and we generated lots and lots of mouse data. We chose 6 doses ranging from 0.1 to 15 micrograms of H56 plus a control group. We kept the adjuvant the same dose throughout. We gave 2 vaccinations, one at day 0 and day 15, to mirror the human clinical trial, which was a 2 vaccination regimen. We took 8 time points sampled over 56 days, and we took 5 mice per time point, so it was quite a lot of mice in the end. And you can see in the graph here, I've got time on the bottom axis and interferon gamma response on the y-axis, and the panels are for the different doses. So you can already see by looking at the data that there is a difference in response ranging across dose. So actually, for the first piece of work, we looked at the data from another angle, and we looked at how the dose response curve looked, and then we took time as the separate panels. So we fit a saturating curve and we fit a peaked curve, which was actually a gamma PDF function, to the data sets for these 3 different time points, and we found that the peaked curve better described what was going on in the dose response. And using this peak curve, we could then predict what we thought the best dose would be. And according to the end time point, which we kind of equated to more of a memory response, something that you might be interested in looking at in terms of being protected, we found that the predicted dose was actually a lot lower than has been tested in mice before. So we established that actually there's something going on here with the immunological response to dose that means that the higher doses are not giving you a high interferon gamma response. So that was our first stage for our first piece of work. So the second piece of work was then to translate these responses to humans. So just as a reminder of what the data looked like, because now I'm going to switch to longitudinal ways of looking at it, this is the data that I showed you before. We actually grouped them together into 3 different groups. So the low group, which was 0.1, 0.5 and 1 microgram, a middle group, which is the 5 microgram group, and a high group, which was the 15 microgram group. And so in order to be able to predict what we thought was happening in the human dose response curve, we needed a human data point. So we had data from phase 2A immunogenicity trial in South Africa. This data here shows the interferon gamma response over time for actually both the H56 trial and H1. We pulled those 2 trials together so we had more data. So we ended up with actually 16 participants as opposed to, I think it was 8 originally. But importantly, in these trials, it was a 50 microgram dose. So the dose was quite high in both the H1 and the H56. So if we wanted to be able to predict how the mouse dose response would look in humans if we were to give them the same range of vaccines, we need to be able to link the 2 together somehow. And so we introduced the allometric dose mapping. So here we assumed that the middle dose group in the mice, which was 5 micrograms given to mice, was equivalent to the 50 microgram dose given to humans. So we assumed that there was this scaling factor between mouse and human of 10. And so with using that scaling factor, we could then say that the response that was happening in the low dose group, which was 0.1 to 1 micrograms in mice, would then be equivalent to 1 to 10 micrograms in humans, and then the high group would be 150 micrograms in humans. So the model I used was just of interferon gamma secreting CD4 T cells, because that is the data that we had, the early spot data we had. The model is very simple. It's two compartments. We start with the recruitment of transitional effector memory cells. You can see going from left to right, the arrow going into the TEM compartment is the recruitment rate. Then these cells either transition to become central memory cells, resting central memory cells in the host, or they die. So this is after 1 vaccination. And then if you were to revaccinate, you would introduce this replication rate, which you can see is the dotted arrow on the side. So the resting central memory cells would replicate, and then after a certain amount of time, they would transition back into effector memory cell compartment and eventually end up as central memory cells again. So this was a model that we calibrated, and actually to calibrate to the early spot data, we counted both the effector memory cells and the resting central memory cells, because the early spot data we thought would be both these cells contributing to an interferon gamma response. So both sets were included. So the results. I'll show you first the mouse data, and I'll talk you through what all these lines mean. At the top, we've got the low dose group. At the side, you've got the early spot, and along the bottom is time. In the low dose group, the blue line corresponds to the median. The blue triangles, if you can see them, correspond to the median data. The yellow correspond to the 75th percentile, and the purple, the 25th percentile. The lines are the model predictions, so I'm not sure if you can see, but they do fit through the data quite well. So we concluded that we had a good model that fit well to the data. So that's the low dose group is at the top, middle dose group, and then the high dose group. And you can already see that there's definitely a difference in the model prediction between the dose groups. So what we then did is we looked at how the parameter. We found one parameter that changed by dose, and we looked at the percentage change between the middle dose group to the low dose group. and the middle dose group to the high dose group. So we just took how these parameter changes between those groups. So the next stage was then to go into the human, fit the model to the human data. So if you look at the bottom left-hand panel, it's the same colour patterns for the data. The model fit quite well, although the data was quite variable. And so that's the 50 microgram human dose group. We took the same relationship between the change in parameters that we had for the mice, and we applied it exactly to the human data set, and we predicted the low dose group and the high dose group. So you can see from looking at this that the low dose group response overall is higher, and so we concluded that a dose of 1 to 10 micrograms in humans may be more immunogenic based purely just on the mouse data. Alongside, when we finished this modelling, alongside this, the dose optimization empirical trial came out, and they concluded that actually they found that the lowest dose that they tried was as immunogenic as the highest doses, and that fell within our range of 1 to 10 micrograms. So that was a good validation of what we'd found using the modelling. Okay. So I'd like to talk now about future work. As Tom alluded to earlier, we have this idea of we've predicted what our best dose is, and we have a certain level of confidence of what we think that prediction might be. But can we use modelling, both statistical and mathematical modelling, to tell us which would be the next best experiment to conduct to increase our confidence in our best dose, which in this is the green line, using the limited amount of mice that we can possibly use? So is there a way that we can simulate what this experiment would look like, and how we should place this mice on this curve to get more information about our best dose, but using least resources, which in the future could be a very interesting application for not only in experiments, but also in clinical trials, where we can say, you must give this person this dose, and you can put this person at this dose, and reduce the amount of people that you end up exposing to the vaccine. So that's the idea for one of our next pieces of work. And following on from that, we would really like to try and develop these quantitative methods a bit more, and optimise vaccine dose for other vaccines. I've said antibody-based, but I understand that obviously we need to consider that we're not just looking at antibodies for these vaccines, but this is we wanted to kind of go down a slightly different route, make a different model, so we can expand our ideas. So we've got 3 ideas that we'd like to go with. The first one is to do very, very similar work that I've just talked about, but use published animal dose response data to predict human dose response for Ebola and dengue vaccines. The second idea is then to say, okay, so we've got a mechanistic model that we think works, that we think is applicable to one vaccine, one disease, but we would really like to be able to expand that out to different diseases and different vaccines. So can we borrow parameters from one disease to predict what it might look like for a similar disease? So I've been collecting all the published data I can find for dose responses for dengue, Japanese encephalitis, and West Nile disease to parameterize a model, and then maybe use this model to say, can we say something about a vaccine we want to develop for, say, Zika, which is extremely similar. So that's another question that modelling we think we can answer with modelling. The last one is to use, I say statistical methods, but we haven't quite figured out exactly what method we're going to use, to predict the best dose for multidimensional tetravalent dengue vaccine dose response curve. And what I mean here is, for the dose response that I've been showing you is just for one, it's just for the antigen. But what happens if we want to say, okay, well, we want to range the antigen and the adjuvant? There we have a multidimensional dose response curve that we need to try and optimise. And there's a good example in dengue where obviously dengue has 4 serotypes, and if you design the tetravalent vaccine, then you're looking at a combination of 4 different variables that you want to try and optimise, which seems like a very complicated question. So, but we want to try and tackle it and see if we can come up with some answers. So, yeah, that's another example of our future work. And that's it from me. I just really like to acknowledge all the people that have given us data and really helped us with the immunology, as well as the lab members at LSHTM who worked really hard to generate our mouse data for us, because it was a big experiment. Thank you.